Hello, everyone, and welcome to today's episode of our Discover Molecular Interactions webinar series. My name is Vivian, and I am responsible for scientific communication at Bruker Biosensors, and I will be your host today. So each month we look at different molecular interactions and we discuss different biophysical technologies to analyze them. And the topic for today is antibody binding. But this time we are not looking at the typical antibody binding to the target antigen, but instead we will look at the binding of the antibody with the Fc part to the Fc receptors. And you will learn all about why this is important and how you can analyze it in just a moment. But let me first introduce to you our two guest speakers today. So I start with Ruiyuan Zheng. She holds a PhD in cardiovascular disease research and cell biology from the University Cologne, and afterwards conducted postdoctoral research in organoid-based disease models at the University of Heidelberg. And she now focuses on driving innovations in reagent discovery and also in developing solutions for drug development at ACROBiosystems. Welcome, Ruiyuan. And our second guest speaker today is Mareike de Pascali. Mareike has a biochemistry background and conducted research at Harvard Medical School on the topic of DNA-correlated nanodiscs. And now she's focusing on developing innovative assays that are aptamer-based or also assays for SPR in a multiplexing assay. Welcome, Mareike.
Welcome. I'm happy to be here.
So we will start with a very brief introduction into antibody-mediated immune responses and quality control of antibody-drug functional tests, as well as the reagents needed for drug screening. And then Mareike will take over and show you a multiplex profiling of different therapeutic antibodies binding to different Fc receptors. She will talk about how to measure 64 interactions simultaneously, as well as how these interactions differentiate in their kinetic behavior. If you have any questions during the talk, please feel free to use the Q&A feed, or if you don't see that in the Teams app, of course, you can also use the normal chat function, or you can also raise your virtual hand at the end after the talks in our Q&A session, and I can unmute you and you can post your questions directly to us. And now, without any further ado, I will hand over to our speakers, and I'm looking forward to the episode.
Thank you, Vivian, and thank you everybody for coming. My name is Ruiyuan Zheng, and I work with ACROBiosystems as a product manager. And I'm talking about the empowering therapeutic antibody drug screening today. Oops. Today's content will cover the antibody-mediated immune response, and in the next part, I will be talking about quality control of the antibody-drug functional test. And in the end, I'd like to show you how the qualified reagent streamlines your drug screening. Let's start from the mechanisms of action of therapeutic antibodies. As you all know, the antibodies consist of two parts, the Fab part binding to the antigen and the Fc part binding to the Fc receptors expressed on the surface of the effector cells like NK cells. And primarily, therapeutic antibodies function through 5 pathways. Through blocking, so where the Fab arms of an antibody bind to a ligand or to its receptor in a competitive way, which directly prevent the native ligand and receptor interaction. And they act through signaling, where the antibody function as an agonist or antagonist, either triggering or inhibiting downstream signaling. The antibodies also work through targeting when it carries a drug of cytotoxicity, that is in the case of ADCs, or it carries some particles with radioactivity in the case of radioimmune conjugates. And last but not least, the antibodies sometimes cause antibody-mediated immune responses. And through this pathway, they function. Elaborately, CDC is defined by the Fc part of the antibody interacting with C1q in the serum, followed by the activation of the complement cascade which eventually induces downstream immune responses. And ADCC is mediated via the NK cells activated by the active interaction of cell bond Fc gamma receptor 3A with the antibodies FcR part. And you have the ADCP, which is induced by the interaction of the antibodies Fc part and the Fc gamma receptor 2A on the surface of macrophages, which in turn augments their phagocytic activity. So now we went through the function of mechanisms of function of the antibodies, and now we went through the antibody-mediated immune responses. Hence, we understand that the Fc receptor interaction directly affects your antibody drug function, and it is essential to test this in a drug development phase. The principles of FcR spending test is pretty straightforward. So now we have in hand the antibody drug that we want to screen, and the next step would be finding its complementary Fc receptor, and you test their interaction affinity and activity. And shown on the right panel are all kinds of Fc receptors we offer in ACRO's catalog. And although it's not listed here, but if you're interested, you can also find C1Q in our catalog. Likewise, in the FcR binding tests, we also do the target binding test. That is the fat part of the protein binds to of your antibody binds to its target, and for that, Acro provides all kinds of targets, including some very hard to express transmembrane proteins, including Claudin family and GPRC5D, etc. And we also provide tumor microenvironment and signaling transduction target, and a neuro, that is a product line where you can find in its collection proteins for neurosciences or neurodegenerative diseases. And apart from target, we also provide tool enzymes. And to do such a functional test, you need a high quality testing reagent. And actually, to test protein interaction, there are several different options of methods. And today, we're talking about the SPR, the Surface Plasmon Resonance. And I'm pretty sure Mareike will cover this part further better in her talk, and I will just briefly introduce it. It is a real-time method. It provides kinetics data, and it is label-free, meaning you don't not need to label your protein for that. And a typical cycle of SPR measurement contains association, steady state, and dissociation phases, where the KD is a parameter that is measured during the dissociation phase. It quantifies the affinity between the two molecules at which 50% of the ligand is bound to its receptor, and it tells us the strength of the interaction happening. And come back to the testing reagents. You do not want to have lots of variation, and a highly constant KD among different lots present high reliability. And like shown here, these three panels placed side by side are the SPR result of three different lots of immobilized human Fc gamma receptor 2B binding rituximab, that is a marketed drug. And they have, if not exactly the same, very similar binding affinity of 10 micromolar as determined on SPR. And besides SPR, we of course also use SDS page to verify the accuracy of our PROTACs, and we also use SecMos to verify the purity of our product. And ACROBiosystems has a quite broad portfolio, but apart from all other reagents and services we provide, recombinant protein development remains one of our sharpest edges. And besides the Fc receptors, which quality I just showed in the previous slides, ACRO provides in its catalog and by customization a wide range of recombinant proteins, among them the multiplex transmembrane proteins. We produce those full-length transmembrane proteins with high bioactivity with three platforms. The detergent micelle platform, virus-like particle platform, and the nanodisk platform. And the detergent micelle platform is recommended for performing the SPR and BLI assay. Indeed, it is quantifiable with high purity, and that allows it to do so. But let's put ourselves in the shoes of a very ambitious drug developer. Let's say we have just very adequate fundings. And does that mean that we just stick to detergent micelle and that's the best quality and best everything for every assay? I would like you to look at this panel. That is a mouse immunized with Claudin 18.2 produced either on the VLP platform or on the SDN platform. And you see that despite, yes, you see effective antibodies specific to Claudin 18.2 was identified by proteins produced via both platform, but you also noticed this difference in the DDM and VLP signals. And this is due to, if you look at here, when you produce those transmembrane proteins with DM platform, you have both extracellular and intracellular domain exposed. And that is why, as a result, the antibodies produced include both ICD and ECD targeting antibodies. And this is to say that it is essential to choose the expression format according to our needs, is because understanding the difference between platforms is also a crucial part of raw material quality control. As mentioned previously, there are a lot of therapeutic antibodies work in a competitive binding way to block the natural occurring interactions between ligand and their receptor. And at ACROBiosystems, we have this TR-FRET inhibitor screening kit, which acts in a same mechanism. For instance, this TL1A-DR3 inhibitor screening kit in its mechanism is when you have your candidate antibody, it binds to the TL1A competitively, and when it does, the DR3 cannot bind to TL1A, and hence you don't have the the FRET signal. And TR-FRET is a high throughput method with no washing steps. It is very easy to perform, so you just mix everything and incubation, and then you're good to go. But it's not real time like SPR. And the kit, like shown here, the TL1A-DR3 inhibitor kit is validated by drugs against TL1A, like afimikibart or tulisokibart. Sorry for my pronunciation for that. And the FRET signal gradually decreases as their drug concentration increases. So, in summary, today I went through different quality control of method of different raw material quality control methods. And apart from our core product services, ACROBiosystems also provides all kinds of kits, including some ADC conjugation kits and ADC internalization kits, and cell lines that can also use in the early drug screening, like the overexpression cell lines and reporter cell lines. And we also have other products and services which I will encourage you to browse on our website. And I hope my talk will be an appetizer for you, and your appetizers are all increased, and we are ready together to look forward for Mareike's sharing. Thank you.
Thank you, Ruiyuan, and also thank you, Vivian, for the introduction. So, as introduced by you, I will not go again through this because you introduced it very well, but I want to emphasize again that we are looking into the interactions of the Fc receptors of the therapeutic antibodies and also what kind of effects that has for the immune response. So, why do we look at kinetics at all? So, the dissociation equilibrium constant can derive from very different interactions. So, as an example, if we have a KD of 100 nanomolar, this could result from a very fast association rate and very fast dissociation rate, as well from a slower association rate of two molecules and a slower dissociation rate. So if we would determine these by a titration curve, we could not distinguish them. But underneath, there were different processes, and especially when it comes to therapeutics, the half-life times are very important on how long two molecules stay bound together and could have an effect. To also show this here on a more applied example. So you can see here the off rates of two different antibodies and their correlation to their IC50 value, meaning that a slow off rate here resulted into a high efficacy. And also the half-life times, depending on the mode of action, here you can see that the T cell activation efficiency is dependent very much on this optimal half-life time. So therefore, we look not only at equilibrium constants, but also at the exact association and dissociation rates. So how do we do this? We measure on surface plasmon resonance, and this is based on the change in the reflective angle. So whenever a molecule binds to another one on the surface, the reflectivity is changed, the angle, and therefore we can resolve the kinetic sensograms in real time. So the measurements itself were done on the Triceratops SPR 64 instrument, which is the newest instrument from Bruker Biosensors. And it's very valid and has a high value in its high throughput. So today we will focus on affinity and kinetics, but of course also different application like screening or epitope binding due to the dual or several injections can also be done with this instrument. But back to why we're here, the interactions of Fc receptors. So the name 64 comes actually from the rotatable manifold. So this SPR instrument does not have any cartridge, but a manifold with 8 channels that can be placed in a vertical orientation to capture a target. So in our case, this would be 8 different Fc receptors, where one would be captured along each injection direction here. And then we can rotate this manifold by 90 degrees and have the horizontal orientation, and then flow 8 different analytes in the horizontal orientation over all the 8 immobilized targets on the vertical direction. So this would then result into 8 by 8 individual measurement interactions, therefore the name SPR64. Additionally to the 64 sensor spots where we measure interactions, we have also interspots that we use for referencing. So that we do not use up any sensor spots for referencing, we have surfaced before and after every measurement spot to have the perfect real-time reference here. In an assay format, we have different modes, but what we measure with the Fc receptors is the multi-injection cycle kinetics, so MIC, meaning that we did the immobilization in the vertical direction, we rotate our manifold by 90 degrees, and then we do injections of 8 analytes in the horizontal direction in increasing concentration. And this goes all parallel. So in one experiment, we can resolve 64 individual kinetic interactions. From the samples itself, so as mentioned many times, we looked at Fc receptors and eight of them. So I will now refer to them with their name of the cluster of differentiation, because I think on a small screen it's easier to see it at once. And of these different Fc receptors, some have very known characteristics that we wanted to probe, but also we wanted to see their effect against different therapeutic antibodies. So we looked at the set of different therapeutic antibodies that had a wild type Fc part from the IgT1 class, but also some that were different. So for example, obinutuzumab has a glycoengineered Fc part, and nivolumab is also a very special therapeutic antibody because it has a different IgT version. This is not IgT1 as the other ones, but is IgT4, and why we will come to later. So from the assay setup, we did repetitive captures of our Fc receptors over a histag in the vertical orientation. And for every kinetic interaction with the antibody later on, we regenerated the surface and captured the receptor again to have like a fresh and unbound Fc receptor for every interactions. And just to point out the reproducibility of the captures over the whole experiments, you can find this in this table, and we used very little sample to do so. So after the Fc capture, we rotated our manifold by 90 degrees, and then we injected 7 different therapeutic antibodies and also 1 negative control parallel at increasing concentrations in the horizontal direction over every one of these 8 different Fc receptors. So this was run in parallel and resulted into 64 individual kinetic sensograms that you see here. So this was done within 12 hours. We basically got all the information of every interaction with everything. And from this one here, I want to point out a few things, but also go into detail more about some specific interactions. But to point out, okay, first the negative control, as expected, we did not see any interaction with the Fc receptor. The obinutuzumab, this is the therapeutic antibody that is glycoengineered, and it's glycoengineered because it should have and result into a higher immune response. And you can already see by one glance the response units for the association and the dissociation towards different Fc receptors is very high and for this antibody. And I also want to point out a very interesting thing that you can already see from this overview is that 2 different mutations of the CD32, the AH, here has a higher affinity, and then the mutation here just does not lead to any interactions anymore. Okay, but now it's more detailed. So I picked here the nivolumab therapeutic antibody because the nivolumab is based on the IgT4 subclass, and it's based on this because it's used in immune checkpoint therapy. So this means that if a patient has cancer, the cancer cell can bind to the PD-1 receptor of a T cell, and the cancer silences the T cell so that the T cell cannot attack the cancer. And in this therapy form, nivolumab is used to block this interaction. So it binds also to the PD-1 receptor, but it does not silence the T cell, but just keeps the cancer cell from silencing this one. And this is, of course, wanted because then the T cell can go into action and destruct the cancer. But of course, the Fc part of this antibody should not activate the immune system, because if the immune system would be activated, then it would recruit, for example, the complement system, and then the complement system would destroy the T cell, but the T cell is very important in destruction of the cancer. So this is exactly what we also looked here, and you can see in the sensograms that the nivolumab Fc parrot does not bind to the Fc receptors apart from the 32 BC with a mid-micromolar KD and the CD64. And the CD64, it binds way stronger with about 20 nanomolar. And this is because of the special composition of the CD64 receptors, which I later on will go in detail, but this is also expected, but also very important test to really see if your therapeutic antibody does what you expect from it. Another thing I want to highlight is different versions of the CD16A, which occurs among the population in two versions. So there's the 16AV and the 16AF. And dependent on which Fc receptors people have on their NK cells, the kinetics behind them are also different. So what you see as results in a rate scale plot here is the 7 different therapeutic antibodies, so without a negative control, the association rate, and also the dissociation rate for both of these receptors. And for all of them, so for all therapeutic antibodies, the dissociation rate is way slower for the 16AV compared to the 16AF. So a very important, valuable information that depending on which Fc receptor in the population you're targeting, you may also have different responses and also immune reactions here. The last thing that I already briefly mentioned is the CD64 receptor. So I picked two sensograms from pertuzumab binding to the 64 and to the CD16AV. And there are different associations and dissociation concentration dependent. And what you can already appreciate by eye is that for the 16AV, the dissociation is way faster compared to the CD64. But to also put this into rates, here you can see on the right side that these are 7 individual interactions of all the 7 therapeutic antibodies we tested against all the Fc receptors. And in red, you see the interactions with the CD64. And here in light blue, the interaction with the CD16AV. So you clearly see the shift in the dissociation that is way faster for the 16AV compared to the CD64. And this increase of the complex stability is because of the nature of the receptor, because the CD64 is the only Fc receptor that has 3 extracellular Ig domains. And because of this additional domain, compared to all the other receptors who only have 2 domains, the binding affinity is way higher and the off rate is slower for this one. And in the context of biology, this is important because CD64 is, for example, on macrophages, on dendritic cells, and they need to detect very low levels of antibodies also to have a very strong immune response to infections. And if you remember the different subclass of nivolumab, you can also appreciate this here, that nivolumab is the one of all these antibodies binding to the CD64, which has compared to them the fastest dissociation rate because of its different IgG subclass. So I hope I could show you how valuable kinetic informations are in also the correlation to their biological function and here immune response. And these 64 kinetics were done in one experiment in 12 hours with relatively little sample consumption on one sensor. And I want to thank especially also ACROBiosystems for this collaboration, but also many other people. Without this project would have been possible. And also thanks to you for listening.
Thank you very much to our two guest speakers. Thanks a lot for these great talks. So yeah, the floor is open for questions. Please feel free to post any questions in the Q&A feed or the chat, or raise your hand and I will unmute you. And I will start with the first question for you, Mareike. What is your next step now in this project? What would you like to look into next?
So as Ruiyuan already introduced, there are many FC receptors that they offer, and one FC receptor that I did not present yet is the natal FC receptor. And this one is also a very special and interesting one because this FC receptor binds depending on the pH, because it transports antibodies, and then depending on the pH, it binds them or releases them. So with the 64 instruments, I didn't show it yet, but because of these 8 channels, we can attach 8 different buffers to them. So we can screen 8 different pHs at one time. And this is the next step in this project.
Sounds great, especially if you think about also other uh, anti like oncology treatments, where you think of the tumor microenvironment having a much higher acidity than the healthy tissues. I think there could be many applications for checking different pH systems for different therapeutics. And then when you think about the rates, like obviously the Triceratops is not the only SPR instrument out there. Is there any comparison data as to how the rates would compare to other SPR technologies?
So there is an application node who did also a lot of kinetics, and of course we look to these as well. And I put them into a rate map to have a visual comparison of them. And what is the same is the kinetics of the trastuzumab binding to 3 different receptors. And of course, the CD64 has the slowest off rate, but in these 3 colors, you can see 3 SPR systems from different providers, and I think everyone can appreciate that the rates really align nicely between all of them.
Thank you.
Thank you for
And then
sharing, Heike. It's really just want to congratulate you on this very nice, beautiful data, because from the raw material provider side, it's always the best to see, when the products is used and run on such, strong capacity SPR machine.
Thank you. Yes.
there's a follow-up question on the buffer system. How many different buffer pumps does the instrument have?
So you can attach 8 buffers for the 8 channels, but of course there are also different pumps that you can also attach on the side a regeneration solution. For example, if you have a very intense wash with high volumes, you can also put 2 reagents additionally, and there's also a water port here that you can really run this in high throughput. I also didn't mention the robot because for now this was not needed, but this instrument is really equipped to do high throughput measurements.
Thanks. And then maybe can you comment from your experience how troublesome is the maintenance of the system? There was a question about the maintenance of the SPR system. Are there any specific steps, or what would you comment on that?
So this system does not have any cartridge or so that you would need to change because of the easiness of the manifold itself. And the only thing you have to do, like with every other instrument, do a weekly cleaning routine that takes about 1 hour with some like normal washing solution.
That doesn't sound too bad. Okay. Can you also comment on how you can control the temperature in the sample rack?
So the sample rack here has 4 plate positions, and to these can be applied an external cooling system that you also attach here, and then you can temperature control them. But this is attached here, and it's made like this that you can still operate with a robot on the side and reach into the instrument, that it's not a fully closed fridge-like system, but the cooling or temperature ring of the plates comes from like below.
All right. Ruiyuan, maybe a question to you. We've seen a lot of human Fc receptors now in use, but I think that's probably not the limitation of your offering. Do you also offer other species Fc receptors if people are interested in doing like mouse experiments or something like that?
Surely, we have these kind of FcR receptors also come in mouse, and I believe also I've seen other species as well to incorporate with people working with different animals.
Thanks.
Thank you.
And maybe another question about the experimental setup. Can you comment on the sensor chip you used, Mareike?
Yes, so maybe I actually go to the slide to show it to you. Because I mentioned that I did a capture here, and the capture was done over a histag. And before I immobilized over amine coupling an anti-His antibody to the surface. And I first started out using a high capacity amine sensor, which has a carboxymethyl dextran surface, and this has a medium charge. But then during the kinetic experiments, we saw that some antibodies interacted with our surface, and therefore we changed the sensor type to the low non-specific binding sensor. And this sensor has a polycarboxylate hydrogel and has also a low charge density, and then this completely eliminated all the non-specificity. So we changed like to the low NSP sensor here.
Thank you. That's also always nice to know that there's plenty of options to optimize your sensor surfaces and so on. So I think that's a big advantage of the technology too. There has been a follow-up for the temperature question. So it was meant, the person was talking about the temperature of the multiplate tower that we can add to the instrument. Do you have any insights on whether that can be temperature controlled?
So actually for the robot that can be attached, you can use any robot you want. We also provide one robot and plate feeder here, but you're very free in your choice, and we also support this here. So depending on what you choose, this of course then might have the capability here.
Great. So I see that our time is almost running out. If you have any further questions or you realize afterwards that you want to know more, please don't hesitate to reach out to us, either by going to our website or by reaching out to the speakers directly. Maybe we can go back to the last slide. I think we had an email address on that one. Yeah, and feel free to check out our website, reach out to us. We also do technical demonstrations. So if you would like to test out the system for your project specifically and see how it works and whether it works in your case, please always feel free to reach out to us. And I hope I see some of you in our future episodes of this webinar series. And you can also go to our website for the previous episodes where we share the recordings also on our YouTube channel. So stay tuned and follow us. And I wish you a very nice rest of your day. And thank you again for participating. And thank you to our speakers.